Thanks a lot, Christina. Thanks, everyone, for coming out. This is going to be a fantastic session. I'm really honored to have been chosen as keynote for this. I'm going to talk to you today about some of my favorite topics, and I'll leave enough time for questions. So, but if there's something pressing, don't hesitate to interrupt, okay? All right, so before I begin, I have to disclose that the biosensor technology that I'm going to talk about towards the very end has been licensed to a company that I started, and I guess I have a 1 out of 10 chance of cleaning up with some money if it turns out to be that 1 out of 10 miracle company. Okay, so my laboratory focuses on this interface of chemistry, biology, physics, engineering, and medicine. And there's 3 basic areas that we work on. One area is we're using mechanical energy to drive protein folding and drive enzymatic catalysis. I'm noticing that my laser pointer doesn't work all that well in this room, so I'm going to try to use this. Just give me 1 second to get it set up. Okay, so that's one area, is driving enzymes, making them go faster, making them fold faster, using shear forces and pressure forces, things like that. The other area is directly connecting biology and electronics, and you'll see a small preview of this, but we also push it down to a single molecule level where we just have one tiny protein attached to one molecular electronic circuit. And then what I want to talk to you about today is using phage display to explore membrane proteins and dissect membrane and engineer membrane proteins, and I'll show you some examples of that today. Okay, so I think this audience is very familiar with phage display. And let me just make a couple of fast observations that will be useful for us later. First, we're going to be talking about displaying our proteins on the surface of these filamentous bacteriophage. And notice the cross-section here is pretty exceptional. It's about 7 nanometers or so in diameter, about a micron or so in length. So these are long, hairy things, and this will come back later. The other thing is that this phage is remarkably, it's the M13 phage used very commonly in phage display. It's remarkably soluble in water. And because it's so soluble in water, it can actually drag membrane proteins out of the membrane and put them into water where they're tolerated. Now, of course, phage display was invented in 1985 by George Smith while he was on sabbatical at Duke University. And it was picked up by many people, but I think a really key advance was made by Jim Wells, where he converted this into a phage-mid system and made it really useful for the experiments I'm going to show you, and actually probably pretty much all the experiments that you're going to see involving phage display at this conference. Greg Winter then showed that phage display is applicable to antibodies, and for this work, Smith and Winter split 50% of the Nobel Prize in Chemistry this year for discovery and innovation in phage display. And the great Francis Arnold split the other 50% for directed evolution. Okay, so today I want to talk to you about displaying membrane proteins. The dogma here is that it's not going to work. Phage display is applicable to soluble proteins, and there are literally thousands of examples of this in the literature. And you know, lots and lots of reviewers told me this would never work, but it does. I had an amazing rotation student who went into our refrigerator and picked out every possible helper phage. And at this point, we were engineering helper phage for some arcane reason that doesn't really have to concern us today. But she went through the refrigerator, picked out all these different helper phages, and found one that would allow display of membrane proteins. And the trick here is that she tinkered with the outer surface of the phage. So if this is the phage over here, imagine now that we zoom in on a small little section of the virus, right where my cursor is pointing. Okay, so if we zoomed in on that, what we would see is an unusual right-handed coiled coil. So with these alpha helical, they look like fish scales running along the length of the virus. And normally, there are 3 negative charges, 3 carboxylate-bearing side chains that are on this unstructured region of this alpha helix that's right on the outer surface of the phage. But Aggie Haidutsky, the graduate student in my laboratory who did this, showed that if you have 1 extra positive charge, suddenly membrane associated and even transmembrane proteins can be displayed on the phage surface. And what she was probably finding is something that looks more zwitterionic, so it looks more like the zwitterionic character of the polar head groups of the phospholipids composing the cytoplasmic membrane. This is notable because phage do not have any lipids on the surface. Phage are a non-envelope virus, and there's no membrane out there. Okay, so let me show you that this works, and then we'll talk about an example. So we can phage display caveolin-1. This is a monotopic membrane protein, meaning it juts into the membrane, runs along for a little bit, and then drops back out. And this can be displayed quite readily. Here I'm showing you some negative controls down here. So this is phage without anything displayed. And then this is the displayed caveolin binding to other things non-specifically. And then also this is binding by a flag epitope on the N-terminus to show that we're getting the full-length caveolin displayed. And that's important because if all of you who do phage display know that sometimes you get little fragments of things displayed on the surface, new stop codons get introduced, so we like to have that as a control. I'm also noting that there's some Mac PC issues going on here, but bear with me. I'll try to explain if I notice anything too important. Okay, so we now have this new helper phage that we call KO7+, because it has one extra positive charge. And here it is displaying the monotopic membrane protein that I showed you schematically I showed you on the previous slide the data, and here it is schematically here. We use that special new helper phage also for display of these beta barrel transmembrane proteins. But I would be remiss to say the problem is solved. I want to tell you the caveats, and I'll try to do this throughout. If I tell you about some new technology, I'll also tell you what's wrong with it. What's wrong with this one is that we can't display any of the 7 transmembrane GPCRs that everyone who's interested in membrane proteins wants to know more about. And so we tried. There's a whole bunch of these listed here, these 2, for example, and neither of these can display. In fact, anything that goes all the way through the membrane doesn't just go through and then come out on the same side. We cannot display unless it's a beta barrel membrane protein. These beta barrel membrane proteins, even if they go multiple times through the membrane, 18, 22 times, no problem on the display. We can display those quite readily, either with our new helper phage or the conventional KO7 helper phage. Okay, so I want to show you that this is now good for engineering membrane proteins. And in my mind, the hardest thing to engineer would be solubilizing a membrane protein, dragging it out of the membrane, turning it into something that's very soluble. And I'm going to show you this with this caveolin-1 protein that I showed on a previous slide. So this is a protein that is, again, monotopic, but it also has this interesting scaffolding function where it binds to lots and lots of different cell signaling proteins, inhibits those cell signaling proteins, and then releases them as the membranes become bent or unbent. And that's one of its functions, actually, is causing these invaginations found on the cell surface called caveolae. Okay, so we can display this on the surface of phage, as I've already shown you. We now can then do selections, where we're selecting for binding to the targets of caveolin, in this case an HIV protein, HIV GP41, and then selecting against, using negative or subtractive selections, binding to hydrophobic interaction chromatography resin. So what we're doing there is we're selecting for greater solubility, and we can do this by increasing the stringency in every round. And specifically what we do to increase stringency is increase the salt concentration. So that makes this hydrophobic interaction with the negative selection stronger and stronger and stronger as you go through the rounds. And the idea there is to increasingly pick out the less and less soluble versions and pick out the more and more soluble versions. We then take all the selectants, subclone those into an expression plasmid, and then simply look for the ones that can express a soluble fragment. And we get some little tiny fragments over here, or smaller fragments. Those are dropouts of the membrane. And then we also get some that are full length. And it's a little hard to say, but there's actually a band there that's not present in the neighbors. And we take those and we express them, and they actually retain functionality. And I'm sort of summarizing a huge amount of data here. Okay, so they now retain functionality. They're soluble in water. They express well in E. coli. Problem solved. So we're doing this. And we noticed something really weird in one of our controls. In one of our controls, we notice that our soluble protein, our soluble membrane protein, can now bind to this fragment of GP41 called T20. This was a ligand originally discovered by Trimeris that's used as an anti-HIV entry inhibitor. And here it is binding over here to our target protein. That's unexpected. And we wanted to improve on that. So that then could be a start for developing a much better ligand for affinity to caveolin. So when we think about affinity, it's really a challenge to start with something that's say 40 residues long and try to improve its affinity if you don't know exactly where it's binding. You can't possibly go through 20 amino acids in every position of a 40-mer peptide, for example. So instead, what we did is we applied a technique that we called homolog shotgun scanning, where we make mutations in every position and mutate for either wild type or a very close homolog. This is equivalent to medicinal chemistry, where we're taking, say, a serine or a threonine. So here's threonine over here, here's serine. We make a substitution, so it's either threonine or serine in that position, and then we select for binding. And if the methyl group turns out to be important, then it will get selected for. If it's not important, then we'll see a mixture of serine and threonine. So anyway, we did this, and we started with this thing down here in the gray, and then through this homolog shotgun scanning, we improved the affinity substantially, and I'll show you exactly how much we improved it in a second. So, greater than 1,000-fold improvement. And I should also say that we're doing kinetic-based selections to improve binding affinity. And this is important. So when you do selections to do affinity maturation, you can either do a selection that has thermodynamic control conditions, where you select for binding and you have an equilibrium. So you just select for the highest affinity things that stay around when you wash more and more stringently, and hope that you're getting the stuff that sticks the best to your target. A better way to do selections for affinity is to focus on an off-rate based selection, where you add an excess of competitor target that is not biotinylated, and then you simply remove all the clones that jump off the target that's biotinylated and bind to the non-biotinylated target. So again, you flood in a huge excess of competitor, and then you remove everything that jumps over to this competitor that suddenly appears. If you do that, you can improve affinity quite a bit. And it's notable, this affinity is, again, off-phage affinity that I'm showing you here. And we also have isothermal titration calorimetry that I'm not showing you to show that we've improved affinity 1,000-fold. Okay, so then we improved affinity 1,000-fold, but again, this is not going to be useful as a drug lead because it's still 40 residues long. So we next went to a whole series of truncated libraries, some of which were on phage, some of which were then ported on cellulose using a technique called spot synthesis, and then there's a whole bunch more libraries getting smaller and smaller until at the very end, we end up with this peptide that's 7 residues long and has a 7,500 increase in binding affinity from our starting ligand. And again, we've gone from 39 residues down to 7 residues, greater than 80% of the length eliminated. So again, I think that illustrates the power of homolog shotgun scanning and next off-rate based selections or kinetic selections. Okay, so the usefulness of the membrane protein there was to provide a lead from which we can then select for ligands and improve affinity quite a bit. But what about simply engineering the phage-displayed membrane proteins and then using them directly for bioengineering? So for example, type 1 diabetes management requires continuous infusions of insulin. And I don't think I'm telling you anything you don't know. This has been done for over 100 or close to 100 years. Of course, type 1 diabetic individuals do not produce insulin due to loss of their beta islet cells. And a real challenge for these type 1 diabetic individuals is controlling glucose levels on the y-axis of this slide here into an optimal range. What we find is that even for patients who have an artificial pancreas or an insulin pump, we find that actually their control of glucose levels is quite poor. It's only optimal about 30% of the time. Okay, so here's the way that system works. So the system has a continuous glucose monitor that's constantly monitoring blood glucose levels, and it's talking to this insulin pump and telling the pump how much insulin to provide. Again, this is in non-optimal system, and furthermore, not sensing insulin presents tremendous risk to patients. For example, if the cannula here gets blocked, or the patient is even doing something as simple as sitting down, and then when they're sitting down and it's injecting insulin into the hip, it can pool up, and then the patient stands up and gets this huge bolus of insulin, which, as you can imagine, is very risky for the patient. So what we're doing is, together with a bioengineer, we're collaborating to build a insulin sensor that would then fit under the skin and work in conjunction with the continuous glucose monitor to provide a much more finely controlled and much more responsive artificial pancreas. So specifically, what we're doing is, on this hydrogel at the tip of a fiber optic cable, and by the way, these cables are amazingly small. They can be injected through the tip of a 24-gauge needle. At the very tip over here, though, there's a little hydrogel. It's engulfed in a little membrane capsule, and in there, we're going to have our insulin receptors that will then be responsive to insulin, and upon binding to insulin, have a fret signal that will then turn on or off depending on the presence of insulin. Okay, now why insulin? Okay, so why insulin receptor? What my collaborator, Elliot Botvinnik, the bioengineer, found is that if you simply try to do this with an antibody, or he actually tried aptamers, what ends up happening is the insulin saturates the system and very quickly shuts down, so that it doesn't actually respond the next time a new dosage of insulin arrives. And that's likely because an antibody that's going to be specific for insulin is going to have very, very long off rates. That's one of the ways that we get really specific antibodies. And again, that type of system will get saturated and then not be responsive at the time scale that's necessary for patients who are receiving these insulin injections. Okay, so the problem is you can't simply put the insulin receptor in place of antibodies, because it's large, it's insoluble, it doesn't express all that well. But wait, insulin receptor is a membrane protein. Here's the structure over here. And we've come up with a way for engineering these membrane proteins. So what we want to do is engineer the insulin receptor and use that as a sensor. Now, the insulin receptor also offers some really optimal qualities for these types of receptors. For one thing, it offers touch and release binding. It binds to the insulin, and then the insulin is released. It has fast off rates. Furthermore, this receptor evolved to have not one, but two binding sites for insulin, a high affinity binding site and a low affinity binding site. And for a sensor, that's a really useful property. That means that it can be responsive across a very wide dynamic range, across high concentrations of insulin where the low affinity binding site becomes important, and then at very low concentrations of insulin where the high affinity binding site becomes important. Okay, so we can display the insulin receptor on the surface of phage, as shown here. So here it is displayed on the surface of the phage, and negative control, again, is phage not displaying anything. This then provides the backbone for mutagenesis and selection and engineering. I should say that we displayed various constructs, fragments of the insulin receptor, because again, we don't necessarily care about the transmembrane region, and for this project, we don't even care about the receptor tyrosine kinase region, so this is a smaller fragment of the full-length insulin. Okay, we then did selections. Oh, and I should also mention that the insulin receptor binds to both insulin and also insulin-like growth factor 1, IGF-1. So we did selections for binding to insulin and also IGF-1, and then we compare the results. Just to summarize, a lot of work. Okay, so this is half a year goes by, and a number of different people, including Sanjana Sen and Dr. Bjarne Vincent, who I believe is in the audience, did these selections, and in the end, we end up with clones, selectants that are specific for binding to IGF-1 and then specific for binding to insulin. So this is IGF-1 binding, insulin binding, and I'm not showing you other ones that we have that are binding specifically to insulin but not IGF-1. This is great news. It would be really useful to equip artificial pancreas, not just with responsiveness to insulin, but also responsiveness to IGF-1. Because the pancreas, these beta islet cells in the pancreas, aren't producing just insulin. They're also producing other proteins as well. And the pancreas responds to these other proteins as well. So for example, if I start talking about chocolate and all of its deliciousness, I will certainly start salivating, and I imagine some of you will too. And that's because appetite hormones start getting released. And you know what? Insulin starts getting released in anticipation of that happy day when the chocolate arrives. So, or when the glucose arrives. So equipping these machines, these delivery pumps with responsiveness to hormones beyond just beyond insulin and even beyond glucose is going to be really important. That's where we're going in the future. Okay, now we have peptide. We have phage-displayed receptors that are specific for insulin. We have receptors that are specific for a whole bunch of other proteins I'm not talking about. What about just sensing directly with the phage? And so this is a collaboration with electrochemists that we've been working on for a number of years. Specifically, what we've been doing is wiring our phage directly into electronic circuits. And we have a new type of sensor that we're calling a virus bioresistor. Here's the way this works. We have 2 gold electrodes, and they're separated by 1.5 millimeters or so. And then we have a layer of a conducting polymer, PDOT, down here. This is a thiophene organic polymer, conducts electricity. And then up here, we co-polymerize viruses together with this conducting polymer. And it turns out the polymer in the co-polymerization becomes oxidized, so it's positively charged. And do you remember earlier I showed you how the virus was negatively charged? The two of these stick together exceptionally well, and we've put these in continuously flowing buffer for days upon days. The phage stick around really well because it's a high avidity interaction between lots of positive charges, lots of negative charges. Okay, so here's our phage in this virus bioresistor. We can then do measurements in buffer and actually even in urine and sera and other biofluids. And then upon binding to the analyte, and this analyte is HSA, this is an albumin, a serum a protein associated with chronic kidney disease. Upon binding to this target analyte, we see a difference in impedance that's measured by this schematic ohm meter shown here. So we can actually quantitate exactly how much analyte is in solution by the electricity going through this virus conducting polymer layer. Okay, now it turns out if you start building sensors in your laboratory, almost immediately you run into all kinds of engineering challenges. And I was surprised at how hard it was to take a sensor that works great in my laboratory, that works great in the hands of 1 or 2 graduate students, and then hand it off to a manufacturing facility to make large numbers of this in a consistent manner. This commercialization and manufacturing is way harder than I thought. It actually took years. But the key was, and I'm not pretending like we discovered this, this is just good engineering, is at every step enforce a process window where you have some quantitative metric for what you need to see before you take things on to the next step. And that's something that we now do in our laboratory pretty routinely, and I highly recommend it as a way of ensuring high quality and consistent quality. So along those lines, when we report data in this area, what we're really looking for are low coefficients of variation. These are sensor-to-sensor measurements between different sensors that, again, are not being manufactured. These are things that are being now synthesized in my laboratory. So each one is its own little art project. And getting down to low coefficients of variation is a big deal. Just to show you how low these are all below 20%. The FDA asks for less than 20% for something that's going to be approved as a diagnostic. And these are controls on the left side over here. Okay, so we can detect HSA at high concentrations. This is nanomolar over here. What's really useful about this, though, is the speed that we can detect things in solution. Do you remember earlier I talked about how the phage were long and skinny? It turns out that when they're arrayed on these surfaces, they form little hairs that stick up and wave around in solution. And we know this because we've done these quartz crystal microbalance experiments that allow us to measure motional resistance, that allows you to measure how far things are off the surface of the electrode. And it turns out that the phage, being far off the surface of the electrode, can grab onto things in solution very quickly. And that's shown here. On the right, this is time in seconds. And in red, that's virus with nothing displayed. This is a control for HSA. This is the cow version of HSA. This is the bovine version. And this is the response that we're measuring at these different concentrations. This response is less than, let's say, 30 seconds to be on the conservative side. But really, actually, most of our measurements is less than 10 seconds for response. And again, that's because the viruses are breaking off the ordered water layer that's found that forms a boundary at the electrode surface. So at surfaces in biofluids, you often have these ordered water layers that slow the process of molecules getting down to the surface. Okay, so this works consistently with lots and lots of different sensors. Oh, and again, this is fast. This is, I think, 10 times faster than any reported sensor. Okay, so we could do this consistently with lots and lots of different analytes because we just replaced the virus. And so the virus is what's being replaced in these things. And furthermore, we can massively improve the sensitivity by controlling the ratio between those two layers of conductivity. So basically, driving the electrons into the outer surface means that we can get much higher sensitivity. And now I'm showing you sensitivity that's in the picomolar range. Let's say sub-100 picomolar range. And again, these percentages are the coefficients of variation, and these are exceptionally low, especially since we're not manufacturing these yet. Okay, let me summarize what we've seen, and then I'll leave you with what I think could help your own research. So what I've demonstrated is that we can successfully display membrane proteins on the phage surface. So the dogma is dead. Peripheral, monotopic, beta-barrel, integral membrane proteins can be displayed quite readily. I've then shown you that we can engineer and dissect these using phage display and protein engineering. And then in addition, we're directly wiring these into circuits, electronic circuits, to allow immediate sensing of biomarkers in solution. Okay, so I always like to leave a talk by telling you what I've learned that you actually might be able to take home and apply yourself. Here's what I've learned. To improve affinity the fastest, first start by choosing a productive area of protein diversity space. It's very hard if you don't have a productive area of protein diversity space to do affinity maturation. And that's why we like this homolog shotgun scanning, where you have a wild type and a close homolog in every position. Kinetic control is a really powerful way to improve off rates and improve specificity. Finally, in terms of engineering, driving for robustness early in the design process is really key there. So thinking early on about manufacturing and process windows will make a huge difference down the road. And then I really would like to encourage everyone in the field to report coefficients of variation for sensor measurements. This is the only way that we can compare different laboratories and know really whether or not something is commercializable. All right, let me end by thanking these important people who made this work possible and the funding agencies. The work I told you about today was done by the names that are highlighted in white. In particular, I'm really grateful for Sanjana Sen on the insulin receptor work, Bjarne Vincents, Dr. Sudipta Manjumdar, a senior research scientist, and Ayesha Attar for the virus bioresistor work that I showed you. Okay, let me stop here and take any questions. Thanks.